start right off. So first, since this is not a familiar crowd to me, I want to introduce myself a bit. So I got an undergrad in pure math and did statistics PhD in theory and empirical processes, and then information theory, and then moved more and more into applied work. Now my group, like 15 people, do both. I think more applied work than theory, but we're coming back to theory about random forest and also deep learning because we've been using it. So for me, data science have three components: computer science, math and stats, and domain knowledge. And machine learning is the popular part. And there's lots of the problems. I have my contribution to single cell is this slide, right? I haven't worked on single cell data, but I hope what I say will be generically useful. So I think what my group does can be characterized as scientific machine learning. What do I mean by scientific machine learning? Is basically use machine learning for scientific discovery, but also build scientific principle in machine learning algorithm and iterate between two steps, and the result should be subject to scientific standards. So since 2013, I have been really pushing for this stability consideration, first with models, data perturbation, and model perturbations, and now develop into also for hypothesis generation for follow-up experiments. And later, we felt like stability actually is a paramount consideration for the whole data science life cycle. So for us, now it's a process. It's not just modeling. You need to think about linguistic stability when you have multidisciplinary team work together. Does matrix mean the same thing to different members of team? And what data do you collect? There's stability there, because if you collect different data, you might should you reach different conclusion about the same phenomenon. And the big underappreciated fact is that data cleaning. It's a huge part of what we do, but we don't talk about it. We just pretend it's not there. I was talking to Olga. I agree with her. Like it's 80% of effort. We don't talk about it. So if you use two different data cleaning methods, would you get the same result? We pretend it's not there. So this is a stability issue. And also, when you do visualization, how do you choose? Which plot do you do? We can really fool people with different visualization of the same data. When you go to the modeling stage, that's where I started. You have different ways of perturbing the data. Even in the IID case, independent identity distributed, you can do bootstrap, can do subsampling, can do cross-validation. And you also have PDE data. We hear a lot about that's really synthetic data. How do you integrate that into the whole data science pipeline? And if you do supervised learning, you have many, many choices. So all of that usually get cherry-picked, and we pretend we didn't try other things. So I think all of this should be made transparent. And then for post-hoc analysis, you have even more human judgment calls. We make different calls, and I think we should write it out in documentation. So I put it under this PCS framework. There's a paper almost finished for PNS with my student, Conbeer. So the idea is really, I started with machine learning. I've been doing after information theory. I went into machine learning for 20 years. And then I'm kind of coming back to statistics, try to bring uncertainty through this expanded consideration of stability to two key components of machine learning, which is predictability and computability. And predictability is the basic way to check reality. It's kind of no-brainer we should check reality, but we hear a lot of talks, at least in statistical meeting, people talk about model fitting without saying, does it capture reality? And then predictability and stability really embed two very important scientific principles, which are prediction and replication. And computability goes without saying, we all have to compute. But I'm also including computability under the computability, also data-inspired simulations. It means that can you use scientific facts to have simulation model, I think can be more utilized in data science. We're also moving into causal inference and using predictability and stability or PCS for hypothesis generation. So I want to show you three examples of PCS, and some faster than others. The first is something called stability-driven non-negative matrix factorization. We add stability to non-negative matrix factorization, and then we add stability to iterate random forest, and we try to interpret things based on deep learning. So this is a project try to figure out in early Drosophila development which parts of the embryo become which part of the organs. It's called cell's destiny. We started with dictionary learning, actually, because I was working in computational neuroscience, and we end up using matrix factorization. So the data is the blue, right? You put these different genes, image 1, image 2, And you try to decompose the idea posed to us by the biologist was that, can you help us find these pre-organ regions, which the morphology was not there yet. We couldn't tell, but we know the interaction forming these are called rooms in a house. The Drosophila was building a house, and they want to find out where are the rooms, and also the genes are like workers. They're going to team up to build different rooms. So all of these blues are the data. Say gene transcription factor 1 and 2 and N. We have some replicates, sometimes. And then we try to say that if we want to decompose, because I was doing computer neuroscience, this sparse coding idea was in my mind, we want to find parts. Let's just do discrete learning. Later we did non-geometric factorization, and we actually find what we call principal patterns that are very, very meaningful biologically. They are like pre-organ regions. And our contribution to methodology is really using stability to select how many components we want. Amazingly, we didn't put any spatial contiguity to force the principal patterns to be continuous. But it turns out to be the case, because that's the basic unit. The genes work on each other. The rooms are, like spatially connected. So we basically use stability to say that if this is a non-convex optimization problem. If we choose different initialization, you end up with permutation invariance and like a sign invariance. That you end up with different principal patterns. We match them through correlation and say we want to find the number of components that's stable across different initializations. And we end up with very clear 21. But also this stability analysis, we also investigate 22 and find that that's splitting one of the component into two, which was too small, talk to scientists, and we end up with 21. So we did a lot of stability analysis. And that's the patterns we got, the pre-organ regions. It's kind of nice, horizontal and vertical, anterior and posterior. Right, so this is a little better than dictionary learning, but it's very stable across the two methods too. And one validations, we were able to reconstruct 10 up to the 11 links in the gap gene network, Bill already mentioned. And for the link we missed, we are just a little lower. So how we did that, we basically used the principal patterns to decompose the embryo to different kind of overlapping parts. And then we did the local correlation. And then that's how we recover the network. Another validation we did prediction too, I will mention, we also want for some theory that what really drives this decomposition. We couldn't do non-geometric factorization, so we went back to dictionary learning. And we actually have a complete characterization of the so-called phase transition. Means that if you have this Gaussian Bernoulli model, square case, we have an underdetermined case, so overdetermined case, so we can map into a square case. That for the simple case that if your dictionary is nice, the D you have is actually just 1, 1, 1, and with correlation fixed, mu off. Then you have this yellow boundary saying that if you're below the boundary in the dark area, then your true dictionary will be a local minimum of the optimization objective function. And if you're above that, the probability will be less than 1. And the other bounds really hug our precise characterization. And the second project, well, one of the other major project we used stability was really with Ben Brown from IBNL, and Basu now with Cornell, our postdoc, and Carl's now UCSF postdoc. And so it's really try to take advantage of another very important supervised learning algorithm, which is random forest, and try to make it interpretable. So Ben had been working with random forest before I joined him for 10 years, really like random forest, but it was not interpretable because if you change the data, the path will change, and you don't have stability for interpretation. As I said, stability has to be a prerequisite for interpretation. So what we try to hear is there is not pattern recognition. It means you want to look for a cat. It's pattern discovery. You actually don't know what you're looking for. You look for meaningful information. And we work with Drosophila Lab, so this is the motivation. We know that there's false order interactions, right, in Drosophila development. And we also know that there's something called French flag model in Drosophila development saying that thresholding is important for development. And if you map that, I'll call the analytical fiber of a model, decision tree is a good map. It's really try to capture in the analytical form, not just qualitatively, but quantitatively capturing this thresholding like phenomenon. So we can characterize interactions of biomolecules through this product of Boolean functions. So random forest, hope all of you know about it, is really building forests from these decision trees with this thresholding behavior. And it's really using perturbations. If you look at through the lens of my stability, right? You perturb the data, you do subsampling, you also subsample the features. And the features really give you different models. So it's both data and model perturbation. And then you build decision trees. But then you do averaging. So that's where it differs, how we're going to do it with iterative random forest. So our iterative random forest, by the name, we start with random forest. And then we reweigh the features. It comes with random forest, there's this importance index called using the decrease of the Gini importance index. You can replace other index there. You have other importance measures, so our algorithm will accommodate that. You just have to put it in. So you up and down weigh different features. Okay, so you go back to random forest. After some point, you do decision tree. So you've built a more regularized decision tree because you're not really treating all the features or the genes equal likely. You're concentrating in a soft way a small set of them. So this allows some interaction to show. Because the statistical traditional polynomial interaction doesn't work with genomic data. You cannot just do main effects and you do polynomials. The computation explosion of the dimension kills you, but it just doesn't work. Because a lot of times, the genes, the transfer don't have main effects. It's really the interaction make them important. So this allows you to go for the interactions. Of course, if something doesn't have a signature, you don't get. But it's really lower the floor for finding important characters. So we have all the trees, and then we take advantage of something developed by two statisticians, Shar and Manhausen, called random intersection trees. So it's a very fast way, almost like stochastic gradient. Take one data at a point, we turn each path into a 0-1 vector. And we not harvest the intersection very fast. And random intersection trees allow us to do that. And then we have a outer loop of bagging. So we don't average, we actually look at the path, and we look at the ones that appear a lot. And in our experience, 3 or 5 interactions are pretty much enough. So the difference between the random forest and us and random forest is that we use important measure view the dimensionality, add stability, and we add an outer loop of stability calculation. So this was the motivating example of enhancer activity prediction in Drosophila with cheap assays. And we use a lot of Boolean function simulation to devise the method too. So it's not just using the data, it was very limited. We actually cooked up a lot of Boolean type of functions, and then we asked the algorithm to discover that. So what happens is that we didn't lose, this is proper testing ROC curve. We didn't lose any predictive accuracy, but at the same time, we reduced the characters we're after. So on your right-hand side is the stability plot. You have stability score, and then you have the names of the genes. The blue ones are 3-way interactions, and the red ones are 2-way interactions. So 3-way interaction, my understanding that the technology is beginning, people can look at 3 interactions at the same time. But 2-way interactions, people have done that with traditional technologies. So we went back to the literature because we work with Sue, who's really on top of Sue Seneker. And 80% what we discover actually have been validated in the literature with physical experiments. So that was really a promising result. And for the 3-way interaction, we become a recommendation system. So that's what I meant. We do hypothesis generation. We don't replace experiments, but we hope we can help the experimenters to concentrate the more high-yield interactions, so you will save resource. And the iterative random forest has a new version we call sign iterative random forest. It's not just we give 2 genes interact, we tell you they have to have high level before they interact. So it's a more refined version. And this is the backbone of our Chan Zuckerberg Intercampus Award to really relate cardiovascular phenotype with genomic component. And we do say that we do single cell experiment, but we don't have the data yet. So there's a connection to single cell here. In the future, definitely. And the PCS, we also emphasize very important, which is overlooked, is documentation. We make so many human judgment calls. For me, model is a mental construct and reality. You have to make the gap and make the connection that you have to use natural language, narratives, and codes. And we also have the paper statistical PCS inference, which will take all the perturbation into account. Then you have a perturbation intervals. So not just probabilistic step, because data cleaning can be taken into account. It's just a perturbation. And you might enlarge your interval for that. And that's just our markdown. I'll do very quickly. We also use this for deep learning related work and try to understand V4. And we can now cluster the different neurons with different preference for their stimuli, and also put back to the experiment closed loop. And DeCarlo's group actually already done that. We are having unparalleled path. Our collaborator, Jack, doesn't do physiology anymore, so we lost that. And we also have been doing interval machine learning. I'll do very quick, defined through predictive accuracy, descriptive accuracy, and relevance to a population and to a particular problem. The paper just appeared in PNS. And we have also developed general methodology to interpret deep learning models. For example, for sentiment analysis, we can explain why this sentence is classified wrong, where the switch happened. And we can also say that actually when you predict puck, you sometimes use skates, and they get highlighted. So this is a general methodology. We ran some small human experiment about whether human can tell a good model versus a bad model, and whether human trust. And our methods seem to be ahead. And we also try to use it to do better debasing for the algorithm. So the papers, one just appeared, one should get accepted, and the software. And the last one I want to put in a plug for the book I'm writing with my student, Rebecca, is really try to help people train in science. It's for upper division undergraduate student, beginning graduate student, and also people who have science background and get into data science in a first principle approach. And so look up. I think we're talking to Cambridge and also MIT and Princeton, and try to get a publisher. Thank you.
Pabin? I will start because it's like, oh, sorry.
Hi.
Thanks for the overview. So I'm trying to understand the stability principle. So what you're doing, right, is test. Better, right?
Yeah.
I'm trying to understand the stability principle. For example, in the NMF factorization example, what you do is you run this for different initializations,
yeah,
and then try to see by correlation, some type of index, if you recover the same type of projections, right?
Yes.
Is there some theory to be done there? Because I mean, there's like full Markov chain Monte Carlo where you can really explore the different space, sort of the whole space where you start, but with those random initialization sounds very ad hoc. So could you show some, I don't know, this is maybe asymptotically gives you really some true model?
For this one, we're writing a proposal to understand the stability results, but we don't have for this particular case. So for other marker, we have some separate work to look at the Hamilton MCMC, right? But that's very more theoretical. So
okay, so it's a pragmatic thing essentially. If you have multiple, if you just restart a thing, or the same for neural networks multiple times, you just see what sort of consistently recover. That's what you call then a stable component.
Yeah, so that's your choice to do that. You can also perturb the data, so the documentation. So there are multiple ways. We chose to do this way in particular case, so you document why. I can also perturb data. We did that. We didn't, get very similar result. So it's human judgment call how you choose the perturbation, and you should document it. Yeah, so it would be nice to analyze it. We don't have analysis yet. My group have been following, get a lot of empirical results before we do theories or moving to our theory now.
Other questions? I actually have a question regarding the random forest model as well.
Yeah.
So did you try to combine that with variable elimination and
with
look
what?
Variable elimination? So because from what I understood from your model, when you do the perturbations, you keep the set of features constant. Did you try to reduce the set of features at each iteration?
Well, implicitly we already reduced because we're weighting. Some the small weight one doesn't get chosen. So we did a self-dimensionality reduction, because we are up-weighting some features and down-weighting some features. So implicitly, we reduce dimensionality, but not in a hard way, in a soft way. Yeah, that's the goal. it's kind of through dimensionality reduction, we added stability into the whole process.
Do we have more questions?
You just teased this at the end, but I'm curious with the neural net example where you were trying to give attribution to the predictions. Was that also?
One?
Yeah, was that also based on a kind of stability analysis or some other?
No, this is a separate thing. We could do a stability on top of this. We haven't done that. So actually, we want to do some adversarial attack to the interpretation, because a lot of the other methods are based on gradient. We think we're more robust, but this is just a conjecture. Because this one, we really went into the detailed construction of deep learning to get the important scores. And gradient-based, it's more about the final fitting. But this is just, we have some human experiment to have some indication of that, but this is something we need to investigate.
Do you think that adversarial attacks have a role with some of the more traditional models too, like the matrix factorization or random forests, or is there something about the deep networks that make them different, where adversarial?
You can even attack a linear model with 2 dimensions. It just somehow the community was not worrying about it, didn't try to attack itself. It's about extrapolation. Adversarial attack is about extrapolation relative to the stability of your model. So the problem with deep learning is the problem become more pronounced because you can have 2 different algorithms fitting the same data exactly training area 0. When you move away a little bit from the fitting, they're very different. I call deep learning octopus. It's like too flexible. I think we can put some bones in it. I think a lot of the science mechanism models should help there. Yeah, I mean, linear model is all bones, right? There's nothing. But you can still get it wrong because you can go too far off your data. So we have another project trying to figure out where are the data points.
Thank you, Vin.